Fantastic. Okay, again, so thank you very much for the really kind invitation to join you guys this morning, and also a massive thanks for the extraordinary time that you're meeting today. When you live here in Aotearoa, New Zealand, you're used to getting up in the middle of the night, so it's nice to not have to do that today. Just a little bit more about me. I'll just get my slides working here. There we are. So often here in Aotearoa, New Zealand, we start by seeking to make connections. So I'll just tell you a little bit about my background. So I'm not from here, I'm from Canada. I grew up camping at the base of this what am I going to say, this mountain called the Three Sisters, and kind of canoeing on the Bow River here with my dad. So for those of you who aren't familiar with Canada, this is part of the Rocky Mountains, and that's a huge part of where I'm from. But my tipuna, or my ancestors, come from Germany and Ireland and England and Scotland as well. So, but I grew up in Canada, and I often say that I grew up with my feet in two types of waters. One of these fresh waters of the Bow River near Calgary, but also on the east coast of the country where my parents were originally from, and in a little place called Bucktouche Bay. So I grew up in there in the saltwater and the freshwater, and all of these experiences have fundamentally shaped who I am, and absolutely contribute to how I walk through this world. So just a massive acknowledgement to those who came before me, and kia ora to any of you who see some connection in there. So just little bit more about us as a research team. We have a couple of mottos. One is official and the other is a little unofficial, but I share it so much now it might be almost official. So one is everyone here is smart and kind. Don't distinguish yourself by being otherwise. So we lead with kindness, which is not the same as niceness. And the other one is being a little weird is just a natural side effect of being awesome. So we're definitely a little bit weird in our team, but we absolutely love that. We've also got some very important kind of mantras that reflect today's talk. And one is every day is a school day, which basically means that we're learning every single day. And we look to embrace our mistakes along the way as long as we're learning from them. And then the other one is teamwork makes the dream work. And fundamentally, everything I'm about to talk to you about has everything to do with my team, and that I really have the privilege of leading. So just a massive acknowledgement to them. In terms of a little bit of the research that we do, again, we sort of wear two kinds of hats within the team, and I'll explain why that is. But fundamentally, it comes down to context. So on one side, we do spend a lot of time thinking about the consequences of small population size and a number of critically endangered species here in the country, mostly birds, but not all birds. There is a little grasshopper in there. And then on the other hat that we wear tends to work in the freshwater spaces. So with critically endangered freshwater fish like this kawato that I have on an image, and also freshwater crayfish. But before I launch into why that is, just for just a bit more of a context in terms of some of the places that I might mention. So if you've not yet have the privilege of joining us here in New Zealand just yet, this is where we are. And do let me know if you're nearby. We have the North Island and the South Island, and that nice little red dot there is Christchurch, where I'm currently based. And then just a couple of other important sites that might come up is Chatham Islands, which is out to the east of the South Island, and then Stewart Island down at the bottom of the South Island. And so a little bit of what I'll be talking about today is species that are found across these four islands or island groups. So speaking again more about context, I suspect within this group, I don't need to spend too much time talking about the FAIR and CARE guiding principles, but just in case. We're thinking about how we can do research in a way and share research in a way that's findable, accessible, interoperable, and reusable, but also in the context of the care principles, which relate to collective benefit, authority to control, responsibility, and ethics. And the reason why I don't think we need to spend too much time on this is that members of your own team have been thinking about this, about contextualizing the fair and care guiding principles in a way. For example, if we're dealing with data that's been generated from culturally significant species to Indigenous peoples, that we're facilitating Indigenous data sovereignty and then upholding those fair and care guiding principles. So just again, a massive acknowledgement to Mihi. I might say a few words in Māori. Apologies, it just happens, but I'll try and follow it up with English if that does happen. But just a huge acknowledgement to a lot of the thinking that's been happening in this space within your own communities. In terms of a local context here in New Zealand, we really need to be thinking about something called the Treaty of Waitangi, or the Māori name for that is Te Tiriti o Waitangi. And there's a lovely explainer at the wee link down there at the bottom, but the sound didn't work last time I tried it. So I've just left that for you guys to check out another time. But really what we want to understand about the treaty is that it was signed in 1840. It's one of the critical founding documents for the country. It was signed by the Crown, and it was signed by a number of Maori chiefs, but not all Maori chiefs at the time. The treaty in itself was penned hurriedly in a matter of days. There was an English version and a Te Reo Māori version, or a Māori language version. And there's a number of issues that have stemmed from the fact that those two versions say different things in different languages. And fundamentally, what it comes down to is that upon signing of the treaty, both the Crown and Maori believe that they held sovereignty, in that the Crown felt they held sovereignty across the entire country, whereas Maori felt that they held sovereignty over themselves, and the Crown was holding sovereignty over themselves. And that's really been at the heart of the conflict between the Crown and Maori since literally the day that it was signed. There is a long and sad history of breaches of the treaty that happened literally again within matters of hours and days of the signing of the treaty. And a lot of what we're seeing now in this space within the country is thinking about ways to actually uphold the treaty and to be responsive to what the treaty has to say. And this fundamentally provides the context for the research that we do, because all of the species that we have the privilege of working on are culturally significant to Maori. But what I'm going to do is just talk a little bit about one of the articles. So there are three articles, or there are three articles in the treaty. And I'd like to talk a little bit about Article 2. And Article 2 talks about something called Tino Rangatiratanga. And Tino Rangatiratanga is effectively this idea of self-determination, independence, and autonomy. So when Maori were signing the treaty, they believed what they were signing was a document that said they had the authority to practice and share their culture in their own way. If we look a little bit further into a little bit of the Te Reo Māori, or the Māori language, or what is written here in Māori, in Article 2, there is another phrase there, which is tauna katoa. So the idea is that Māori felt believed when they signed the treaty that they had the authority to control all things treasured. So tauna katoa. So tauna is those things treasured, and katoa is all things. And so we can understand almost immediately how in a colonized country that can go wrong quite quickly. So when we think about all things treasured, this includes things like the Māori language, so Te Reo Māori, what is Mataranga Māori, which is Māori knowledge system, Tikanga Māori, which is Māori ways of doing things, practices, and yeah, just ways of being. And then there's a concept which is whakapapa, which is often translated in English to mean genealogy, but that's a really simplistic interpretation of what whakapapa is. And so I'll talk a little bit more about that in a moment, because again, it further contextualizes the work that we do. So we've again been very, very fortunate to be doing this work for a few years now. And we're definitely seeing further contextualization of the treaty here in New Zealand by the research community. So that includes the genomics research community, as well as the research community in general here in the country. So some of the work that I'll be talking about was done before this was happening or in parallel to when this was happening. But for example, if I am teaching this work now to my students here at the university, we're talking about things like these guidelines for genomic research on taonga or treasured species, and most recently, this best practice guide for conducting research involving these taonga or culturally significant species. But what I'd like to do is provide kind of some tangible examples of the type of work that we've done. And I won't be going through each research project in detail, but rather my intent here is to provide a little bit of an overview. I'll talk a bit about some of the work that we've done with place-based knowledge holders in the context of what we may call local communities. But most of what I'm going to be talking about is Indigenous peoples, but then I'll bring it back to local communities towards the end. So the first place that I'll start is here. So this is a paper that was led by Levi Collier Robinson, which is the fellow on the left-hand side of the slide. And so Levi did his undergraduate research here at the university, and then joined my research team as a master's student, and then later a PhD student. So this particular paper was a product of one of his master's thesis chapters. And Levi is Maori. He whakapapas, or he has ancestry to a number of different tribes and sub-tribes. But most importantly, one of them is our local sub-tribe here. So the land upon which the university sits is a sub-tribe called Aotearoa, and Levi whakapapas or has genealogy back to this sub-tribe. We were also joined by another Maori researcher who was a PhD student at the time, Chanel Toms, a non-Maori PhD student, Aisling Rain, and then one of our kaitiaki, or the guardians that we worked with, which is Makarini Urupene, in this work. And the context with this particular work is that it really framed absolutely everything we did when we started working and generating genomic data. In the past, we were transitioning from using a small handful of genetic markers, mostly microsatellites, and we were moving into using either reduced representation sequencing or later whole genome resequencing. And we would usually generate reference genomes with our work, often what we would call a good enough genome. But depending on the research that we're doing, it might be a bit more high quality than that these days. And the context here is that DNA, so is actually a physical expression of whakapapa. And where whakapapa, if we just think about it as the relationships between people, like we might think about with genealogy, it's not quite right. We have to also think about the relationships of people with place and the relationships of people with their ancestors. And so it provides kind of a whole of system approach to thinking about the connectedness of all things in a living space, and also in the context of ancestors into that space as well. And so the idea here is that what Levi was exploring was that like DNA, whakapapa is unique to any hierarchical group, And so what I do here is actually read what he said because I don't feel comfortable speaking on his behalf. So this uniqueness inherently renders whakapapa and by extension DNA as a tauna, so something treasured, and something that is tapu. Where here tapu is refers to the sacred nature of an object, which means not that you can't touch it, but rather it must be actively protected or managed. And therefore tikanga, which is this way of thinking about um, appropriate ways to engage in things that are tapu and includes practices and protocols and ways of thinking and being. And when we kind of move into thinking about DNA as whakapapa, tikanga or the way we do things should influence how we consider this genetic and genomic data. And one of the most important contributions, I think, of Levi's work was really the emphasis on the fact that new technologies may require new tikanga, and the idea that some may view Indigenous peoples and Indigenous knowledge systems as being something in the past and something very historical, when in fact those knowledge systems are living and breathing and adapting and changing to our contemporary and ultimately our future context. And so when we're exploring ways to think about this DNA, we're very much in the here and now within those contexts. So one of the ways that we uh, went about ensuring that we were actually being tika or doing the right thing when it came to working with the DNA from two particular species that we're working with in this particular project is that we considered four key Indigenous principles. Rangatiratanga is one we just talked about, which is the authority have the Maori have the authority to practice and share their culture in their own way. There's whanaungatanga, which is about building and maintaining meaningful relationships, which I really understand this group is really keen to be thinking about. We have kaitiakitanga, which is about enabling the guardianship of taonga. So that includes, again, the environment, the knowledge, the culture, as well as the species within. And a really, really big one is tohunatanga. And this is pursuing knowledge and ideas that develop capability, capacity, and expertise. And this is a two-way exchange, not in the idea that we as the researchers know all the things and we're gifting them to the recipients, but rather we're learning as much from them as they're learning from us. So that's how we framed that work. And fundamentally, what we're looking to do is to be responsive to all four of these principles throughout the research cycle, starting from the framing of the research narrative, through sampling, through the generation of those data, through the storage of those data, thinking about what that access looks like, and then really importantly, the sharing of those data. So I could talk about this for a long time, but I won't. What I will do is just provide another example and provide additional context to the work that we do. So this is Aisling Rain. So she was a PhD student on the same project that Levi was a master's student on. And we were joined by a number of colleagues, both Maori and non-Maori, in this project, thinking about reimagining conservation translocations, where conservation translocation is simply moving a plant or an animal from one place to another in order to conserve it or enhance conservation outcomes. And part of the reason we spent a lot of time thinking deeply about this is because of our local context here in Aotearoa, New Zealand, in that our legislation actually doesn't reflect the threat status of our most threatened freshwater species, or even our least threatened freshwater species. And this is just by way of comparison. So I've used a bird, and this is a nice big giant weta, to represent our terrestrial vertebrates and invertebrates. And then we have kawaro, this wee fish, I'll talk about shortly, and kekewa, freshwater crayfish, to represent our freshwater vertebrates and invertebrates. And what we can see is that the proportions of threatened and at-risk species across all of these groupings are actually relatively similar when we compare the invertebrates to the invertebrates and the vertebrates to the vertebrates. But in contrast to our terrestrial species that actually benefit from extensive legal protection, there's limited legal protection for any of our threatened freshwater species. So much so that the only freshwater fish that's protected by law here in the country is the grayling, and the species is extinct. So they protected it after we lost it, which is a little bit sad. Um, and what this actually does, this lack of legislation that we can then build from, it actually hinders our opportunity to actually translocate these threatened species in a way that is consistent with best practice. So essentially, when we have extensive legislation that can enable the building up of a really solid scientific evidence base, and then that evidence base leads into translocation protocols that communities, for example, local communities might want to use to translocate terrestrial species. If we move over to the other side and look into the freshwater space, there's very limited scientific evidence in this space. There's very little work that we can go from. And then what that means is that there's no translocation protocols, or they're lacking. So so much so that I recently provided some feedback on our government's first attempt on actually coming up with some of these policies and protocols. Yeah, and I have opinions, but I'll keep those to myself. Um, so what this led us to do was to think about the fact that when we think about the conservation translocation cycle, we might start thinking about our objectives and our success indicators. So how do we know that a translocation has worked? We think about our strategies, we go ahead and do the implementation, and then what we really want to do is we then want to monitor post-release and manage accordingly. But what often happens, particularly for translocations that are led by local communities, is that there's very little incentive or funding to actually maintain a post-release monitoring strategy, let alone iterative management. Often animals or plants are translocated and left, and then years may go by and we go, oh, this failed, there's no animals here, what happened? And then we don't know. Or if it has succeeded, we don't know why it succeeded because we weren't able to monitor. And so one of the things we thought about a lot during this particular project was that most of the reasons why we see Indigenous communities, particularly here in New Zealand, so in terms of Maori communities, the species in a freshwater space that they want to translocate are actually those species that they want to eat. And so, these can maybe called Mahinga Kai species, but that Mahinga Kai relates not just to the harvesting or eating of that particular species, but all of the knowledge and the language associated with that species. So if we're translocating different species that are highly culturally significant, and ideally what we're looking to do is to not just see conservation benefits, but also to enhance sustainable customary harvest, there's a huge amount of impetus and energy in actually that post-release monitoring and the iterative management, because there's a high interest in that translocation working well. So that's part of some of the thinking that we've done. And then related to that, this is another data chapter in Ashling's thesis. Again, we have a larger group of collaborators here from more sub-tribes on the South Island, and some of our other colleagues as well. And Ashling was working on a species called kikewai, or freshwater crayfish. What we were interested in was actually characterizing phylogeographic structure across the South Island. We do see a little bit of a gap in some of our sampling, and the reason why that exists is that we only sampled in those places where we had relationships. So we started, see if I can get up here. So we started kind of up around, this is Canterbury where I live, and then we built relationships down in, this is Otago or Otago, and then down into Southland or Mirihuku. And so all of this sampling was conducted, especially down in the south, down in the bottom of the South Island, alongside Kaitiaki or guardians of those particular subtribes. And one of the things that we were interested in was evidence of past translocation or human-induced translocation, because there's a strong history of Maori actually moving freshwater crayfish. So, for example, if they were moving along this side of the South Island, this is where the greenstone or ponamu or jade exists. And so there are a number of trails that exist where individuals will travel across the Alps to go and source the greenstone and bring that back. And there are sites above waterfalls that freshwater crayfish, for example, could never get to. And there are stories associated with the movement, or knowledge is associated with the movement of those crayfish. There's also big, beautiful, large cabbage trees as well that were used as sentinels, but also for food as well. But just as an example of how, in addition to Indigenous knowledges from our local communities can help us make sense of the patterns that we might see. For those of you with a keen eye on this particular, this is just a structure plot. We're here in Waitaha or Canterbury, and there's one population here that does not look like the others. And this is this site right here. And when it first happened, of course, Ashling came to speak to me, and she's like, "I'm not sure what happened. I need to make sure I haven't swapped samples. This is really confusing." And she had checked make sure that there was no mistakes. And then we spoke with one of our co-authors, a fellow named John Hollows, who's an avid hunter and fisher. And he said, "Oh, well, that's Lake Janet, and that's a human-made lake. And those were big pig hunters that actually brought up crayfish from down south and translocated them into Lake Janet." And so that knowledge instantaneously made sense, and very, very serendipitously, not only a few weeks later, Ashling was in the bush and she came across some pig hunters, and they talked about Lake Janet and the fact that there was crayfish there. And so so it just we goes to show that we really need to be mindful of those place-based knowledges when we're working in this space, particularly for species that are culturally significant, not only to Maori, but also to local communities as well. If we think about how hunters and fishers may sustain themselves when they're in the bush for a fair bit of time. There are also examples if we looked at the sub-hierarchical structure, but I won't show you that. You can have a look at the paper if you'd like. But now I want to switch gears a little bit, and I hope I'm not talking too long. Is that all of this research up until now is on these freshwater species that are not protected by law. And now what we're going to do is we're going to shift to terrestrial species that are protected by law. And then in particular, some of them are further protected because what's been happening over the last 30, almost 40 years is there's been a number of grievances brought to the Crown by sub-tribes and tribes to seek redress or for the grievances that happened to them since the signing of the treaty. So Ngāi Tahu, which is the large tribe that sits across a lot, not all, but a lot of the South Island, signed its treaty settlement with the Crown quite some time ago. And when they did that, in the Settlement Act, it stipulates that for a number of different threatened birds in particular, there would be Ngāi Tahu representation on the recovery groups for those birds. So one of those birds happens to be a species called Kakariki kereka, or orange-fronted parakeet. And who I've got on this image is Stephanie Galla, who was the PhD student at the time, and then Yvette Couch-Lewis. So Yvette Couch-Lewis is the Ngāi Tahu representative on the Kakariki kereka species recovery group. And we've been working together for about 15 years. And the point that we make in this particular paper, and also thinking about what underpinned another one of the papers that came out of Stephanie's thesis, is actually what we did to generate the reference genome for Kakariki kereka. And it was really important to us that Yvette was the person who chose which individual we would use from our conservation breeding program. And so essentially what I've got here is just a little bit from the paper in itself that talks about how we knew we wanted to generate a reference genome from a male. So we thought about some good options. We showed a number of partial pedigrees to a vet, and a vet had a good look through the pedigrees. We called them family trees. We didn't call them pedigrees. And we can see instantaneously that even thinking about family trees or pedigrees is an instantaneous connect to the concept of whakapapa, that connectedness. And so a vet was looking for the bird's whakapapa. And she ultimately chose this boy here, who's Maverick. And one of the big reasons why she chose Maverick is that the conservation breeding program that we have for this species was sourced by individuals from three valleys in the nearby Canterbury valleys. It was the Hawden, the Hiranui, and the Poulter Valley. So Maverick really appealed to a vet because he held the whakapapa from all three of those valleys. Further, he was also a really good dad. He had lots of offspring. So it was those two things that led her to choose Maverick. We went about it slightly differently for another species. I'm now out on the Chatham, so we're east of the South Island of New Zealand. And again, this is intended to be an example of the work that underpins what can be quite a technical paper. So this paper was around characterizing toll-like receptor diversity in some whole genome resequencing data that we had for this particular species, which is called Chiriwat, Tutururitu, or shore plover, and I'll use shore plover. And I'll just tell you a little bit about some of the work that we're doing. So again, we're out on the Chatham Islands, and actually I'm off to the Chatham Islands in 1 week today to do a bit more work, but on a different species. But the story of the shore plover is that what happened, we knew we wanted to do a reference genome, we knew we wanted to do a boy. So Alina, one of my PhD students, chose two eligible males from this particular island, which is called Aotearoa. And what Alina and actually Stephanie did at the time, there are two schools on the main island of the Chathams. They went to the first school on day one, and they said, okay, folks, we're going to choose two of these males, one of these males, to be the reference genome. And so what should we name them? So the kids at that school came up with Kina, which is like sea urchin, and Maui, which is a very famous god in Te Ao Maori, or the Maori world. And then the next day, what they did is they went to the other school on the main island, and they said, okay, your friends down the road, they've named Kina and Maui. Which one should we do? And then so those kids voted, and Maui became the reference genome for that species. As well, some of the work that Alina did as part of her PhD led to a decision to augment our conservation breeding program on the main island for this species with eggs sourced from Rangatira. And so we did a lot of work looking to maintain the connects between our research and the community, because the Chatham Islands is quite far away. It's challenging to get to. So we put a lot of energy and effort into those communications. And this little poster right here represents the 21 eggs that were taken off of Rangatira. And this particular poster shows us images of those birds. A subsequent version of the poster shows which birds were paired with which birds and what facility they were at. So again, these things they underpin the work that we do. We couldn't do the more technical work without having this underpin it. I'm just looking at the time. I think I've talked a lot, so I'll move through this fairly quickly. Have we got enough time, or do you guys need to go?
I think we have another 5-10 minutes to go.
Oh gosh. Yeah. Okay, I'll go quickly because you might have questions. I'm sorry, it's a bit late. I'm losing my ability to.
Yeah, 5 minutes if you want to have some questions. Yeah.
Okay, all right. That I could do that. Again, this is work we're really interested in structural variation, particularly in kakapo here in New Zealand through much of the work led by Jenna Wald, who was my PhD student, is now a postdoc. But much of what we're learning in this space is actually underpinned by the generation of two fibroblast cell lines for kakapo of two chicks that passed away a couple of years ago. And we've done a lot of thinking here in New Zealand around how to handle genomic data for culturally significant species. But now we're moving into, well, what about actually cell lines? What about gametes? What about cryopreservation? What about reproductive technologies that we might use? And so we have a project that underpins this work with a number of our Māori colleagues, including two young Māori researchers. And this has spun off into some additional work led by my Matt Wally and some of our colleagues in Japan, as well as others here in New Zealand, where Matt is really interested in germ cell cryobanking and transplanting for conservation benefit for some of our threatened freshwater fish. So there's a lot of thinking about the methodological, ethical, cultural considerations around the collection, use, and storage of these types of samples. So we're thinking a lot in that space currently. We've also done some work in the data management space. And so this particular team came out of a co-design group here at UC because we've been exploring essentially the e-research landscape here at UC. So in this particular paper, we have researchers as well as professional staff, so library staff, e-research staff that support the work that we do, and basically a lot of the big compute cluster work that we do. And as part of that paper, we have what we're calling the Biodiversity Genomics Data Management Hub that has a series of modules, and essentially the management hub is intended to be the gifts to our past self, the things we would have loved to have known when we were first starting on this data journey. We do have there's a series of modules. One of them is focused on facilitating Indigenous data sovereignty and upholding the fair and careful guiding principles. And then the 10th one is actually the product of my meeting with the ERGA committee, who asked, I think it was Christian asked, whether or not we could have a module 10 working with local communities. So it is very near to being done. I was hoping to reveal it to you now, but I'm glad I didn't because I have no time. But where it will be available very, very soon. But just as a bit of an overview, if we think about engagement with local communities or with Indigenous peoples as a spectrum, we can think about low engagement where it might just be a one-way dissemination, all the way over to co-creation of research. And this is a particular image taken from one of Ashling's paper, where we talk about how critical it is to think about that place-based knowledge, to work with local communities and Indigenous peoples, to make sure that we actually have context when we're interpreting our genomic data. So I'm going to leave that thought with you. And I'm just reflecting on a recent meeting that I was at where folks asked a bit about how we communicate that what we're working with, the work that we're doing. We spend a lot of time talking about puzzles. And so never underestimate the power of a good metaphor. So we talk about genomes as completed puzzles on the top of a puzzle box. We might talk about smaller puzzle pieces for short read data, larger puzzle pieces for long read data. We can then extend that analogy when we talk about coverage. We talk about coverage a lot when we think about the number of puzzle pieces on a particular image or part of the puzzle. That really helps us when we're talking about budgets and options and how many samples and how much coverage, because it contextualizes the work that we do. So this we find really useful. And we also use what we call these one-pagers for dissemination. So I won't expect you to read this, but this is written in a language that's responsive to our local context. There's a lot of te reo in here. The species in itself goes by different names. So if we're talking to Aotearoa up here, we're using KKY. That is the name that they use here. If we're going down south, we're calling these freshwater crayfish kora, and we're using that in all of our comms. So we're looking to be responsive to that. I hope this has been helpful. This is just where I really acknowledge absolutely every human that got me to this space. And I apologize for talking a little bit too long. And if there is some time, I'd be delighted to take some questions.
Thank you very
We
much,
have it.
Tammy. That was very enlightening talk. Very, very nice indeed.